Hi, my name is Renee Chow, and I will be presenting the first portion of this article presentation. In this week's article, we are looking at a role of Wnt signaling in self-renewal of hematopoietic stem cells. Michelle will be going over the second portion of this presentation, and I recommend checking that out after this video. So in this video, I'll be going over the background information, specifically on the hematopoietic stem cells, Wnt signaling, the researcher's hypothesis and main questions, as well as some extra notes that I wanted to discuss before proceeding on to the results, where I'll be talking about Figure 1, Table 1, and Figure 2. So starting with hematopoietic stem cells, which I will now refer to as HSCs, is a cell isolated from the blood or bone marrow that can renew itself, can differentiate to a variety of specialized cells, can mobilize out of the bone marrow into circulating blood, and can also undergo programmed cell death apoptosis. As you can see in this image that I have here, you can see all the possibilities that the HSC can differentiate to. So one such example is the lymphoid progenitor cell that leads to the T cell or B cell. It can also differentiate into the myeloid progenitor cell here and can continue to neutrophil or basophil and continuing on. They are extremely important and are used in many treatments of malignant diseases such as leukemia and lymphoma, and non-malignant diseases such as sickle cell disease. They are also used in clinical trials for treatment of autoimmune diseases, genetic diseases, and more. But the treatment that we are mainly focusing on is known as the bone marrow transplant or stem cell transplant. So we are specifically looking at long-term self-renewing HSCs, which can be isolated by their expression of lineage markers. So here we'll be going over Wnt signaling pathway, and I highly recommend going over Module 10, as Dr. Brothman does explain this and other key points that may be helpful before you read and write about this article. So first we'll go over this image I have here. So this pathway is known as the Wnt beta-catenin pathway specifically, and it's a canonical pathway. And you can see that Wnt signaling is in binary state. So it can toggle between on and off. And the main component of this is the Wnt ligand. So I'll first talk about this on state here. So Wnt ligand, which in the article it's labeled as Wnt3a, it attaches itself onto Frizzled, which I'll show in this off state here that since it's labeled a little better. And it'll attach itself to that receptor along with the co-receptor LRP6 here. So once that attaches, it actually relocates this destruction complex here that contains some key components such as axine and dishevel, as well as a few other ones, APC, GSK3, and CKI, and also this phosphate ion here. And what happens is that this destruction complex is now attached to the cell membrane or to this receptors, and beta-catenin has the opportunity to then relocate to the nucleus and attach itself to the transcriptional factor, which then stimulates the expression of the target gene. So this is in the off state when this Wnt ligand is not available. So here you can see it's empty, and this destruction complex now targets this beta-catenin that's available and phosphorylates it, which then triggers it to degrade. So now it's no longer usable. So now this nucleus is turned off, so nothing will happen in this case. So moving on to the hypothesis and questions, the researchers had many questions about how this pathway would work and all the specific variables that worked along with it. But the main hypothesis that they kind of discussed is that Wnt signaling does have an influence on HSC, but the main broader questions that they asked are, is Wnt signaling required for HSC growth? Does inhibition of Wnt signaling affect HSCs? Does activated Wnt signaling affect long-term HSC growth? Can Wnt signaling regulate HSC self-renewal by upregulating certain genes? And how does expression of beta-catenin affect upstream Wnt signals? So a few key notes I wanted to go over before proceeding on to the results is that the researchers used two kinds of mice, the wild type and the H2K BCL2 transgenic mice. So isolated HSCs from wild-type mice have a difficult time expanding and would require a lot of growth factors in order to keep them alive. So in order to accommodate for that and make sure that no other factors affected the experiment, the researchers used the transgenic BCL2 mice because they only proliferate to steal factor alone, which is a growth factor. But I did want to mention that in the supplementary article that was attached to this week's article, they did test whether or not there was a difference in using the wild type versus BCL2 when determining the responsiveness of HSC to Wnt, and they did not find a significant difference in using either of them. So I also wanted to note that the researchers, in order to distinguish mice from either being the donor or recipient, they use cell surface markers that are always expressed in certain mice. So in this case, we have Li5.1, which is always being expressed in the donor mice. And we have Li5.2, which is always expressed in the recipient mice. So now we've moved on to the results. And here I have the first part of figure 1 shown. And the researchers wanted to see if an activated Wnt signaling pathway would allow expansion of the cell outside of the mouse. So they continuously expressed beta-catenin in order to make sure Wnt pathway was always on, and they used GFP so that glowing cells could be identified if they were transfected. So the researchers fused GFP to both the vector control along with beta-catenin and infected them into isolated HSCs and then sorted them out using fluorescent activated cell sorting and then analyzed those cells. So here in figure A, they are measuring the percentages of cells that were in active cell division, where the x-axis here measures the amount of DNA per cell. So actively dividing cells will have twice as many fluorescents indicated, which it is shown here in these second boxes. So as you can see, vector control GFP positive only had about 34% of its cells actively dividing, whereas beta-catenin with GFP positive had about 58%, which shows that these cells did in fact expand. But to do another test, they also did a very simple cell count where the black is the beta-catenin and the gray is the vector control. So as you can see here, vector control had very little to no cell count over the number of days, whereas beta-catenin actually increased as it continued to day 60, and it almost appears to be almost 100 times more than vector control. So with these two figures here, it indicates that the vector cells had little to no expansion, whereas beta-catenin had a very large expansion, and confirmed that overexpression of beta-catenin will lead to active cell division. So now we've moved on to the second portion of figure 1, where the researchers wanted to know, are these cells still HSCs? And to verify that, they need to characterize the cell's phenotype, and they did it through staining and flow cytometry. So here in figure C, we are comparing the nuclei to cytoplasmic ratio, where as the nucleus shrinks, the ratio also decreases. And as HSCs differentiate, their ratio would decrease because their nucleus becomes smaller. So in this case, you can see it in the vector control that this nucleus is shrinking compared to the actual cell itself. And you can kind of compare that to the beta-catenin, where it's pretty consistent and it's much larger here. So the vector control has a much lower nuclei cytoplasmic ratio compared to the beta-catenin, which indicates that the beta-catenin did not differentiate. And here in figure D, we are comparing the lineage markers after they went through flow cytometry. So we have this black line and gray line in this first diagram, and the black line is indicating the beta-catenin, whereas the gray is indicating the vector control. And HSCs are more likely to be lineage negative, and as you can see with this beta-catenin, it's much more negatively inclined than the vector control here. And you can also see in this last diagram where it is comparing lin-negative, SCA1, and c-KIT, all of which are lineage markers for HSCs, make up 99% of the cell. So this really indicates that the beta-catenin did not differentiate the HSC cells. So now we're moving on to our first table. And the researchers in this case wanted to know whether these cells are still functional HSCs. So to reiterate, the researchers took a mouse, they irradiated it to a lethal level, and transplanted the donor mouse that they isolated to the vector and beta-catenin mouse. They measured how frequently the donor mouse cells were found in the recipient mouse using the markers we discussed earlier, Li 5.1 and Fly 5.2. And here we have where they measured chimerism, which is calculated by the ratio of donor versus recipient cells. So when we're first looking at the vector control here, we're looking at the peripheral blood cell type specifically, and you can see that its reconstitution was at 0, which means that no donor cells were incorporated, and it kind of follows along with the chimerism. Whereas if you look at beta-catenin, where we also compare peripheral blood, B cells, T cells, and myeloid cells, they all had successful reconstitutions, and they had an average chimerism that was pretty high, and showed that they were able to be incorporated and were successfully reconstituted. So continuing on with figure 2, which will be my last slide in this presentation. The researchers used flow cytometry to show in the recipient mouse what percentages of its cells were B220, CD3, and MAC1. So B220 is a cell that's been differentiated from the donor mouse's cell, donor mouse's B cell, whereas CD is the same with the donor mouse's T cell and MAC1 with the donor mouse's myeloid cells. And as you can see, all of these here, if added together, is almost close to 100%, which means that from figure 1, table 1, and figure 2, you can kind of conclude that beta-catenin prevents HSC differentiation, but also promotes proliferation, inducing self-renewal in serum-free medium. So here are my references that I've included, and again, I would recommend checking out Michelle's video for the rest of the results, as well as the discussion in our proposed experiment. Thank you.